Hey everyone, my name is Ella Shiratsky and I'm a postdoc at UC Berkeley in Mary Firestone's lab. Welcome to our lesson 2. Today's theme is going to be all the things you can do in Excel and how to do them in R. You already know this. When we have an Excel table, like the OTU table that I provided here, look at it in Excel, you can do all sorts of actions on it, like you can sort or filter it, you can sum or average columns or rows. You can change column names. You can merge it with another table. And I'm just going to show you how to do all of this stuff in R. So let's start with one of the classic data frames out there. It's called the IRIS database. So we're going to start by loading it. And now you can see here on the right in the global environment, you have IRIS. So let's see what it looks like. str gives you the structure of any variable. So it tells you it's a data frame, it has 5 variables, and each one of them has 150 observations. Now we can view the table by either using the view function or just clicking on it here. But we can also just see the top of it by using the head function. So here is the subset of the first 6 rows of the IRIS database. So you get an idea of what it looks like. 2 really important functions are nrow and ncol, which tell you how many rows and how many columns are in this data frame. So there are 150 rows and 5 columns. Last lesson we talked about the names function. For data frames, there are specific functions that will give you the column names or the row names. So col names gives you the column names, row names will give you the row names, but in this case it's just numbers. But notice that these numbers are in quotes, so they're actually strings or characters, they're not numeric. Now we can change the name of a column. So col names is actually a vector that you can change, that you can edit. So let's say I want to change the name of the second column from sepal width to set width. So let's do it like this. And now we can type down here in the console, col names, IRIS, IRIS, and the name of the column has changed. And then we can change it back. We can go back to the console. Now you can retype the same command, or you can just use the up arrow to type previous commands. So see, now the name of the column is changed back to its original name. Another really useful function in Excel is transposing a table. So rows into columns, columns into rows. So let's do that. That's a function named T for transpose. So now you have 5 rows, and for each one of these rows, there are 150 columns. All right. Note that I just printed this. I haven't actually changed the table. If you wanted to change the table, you'd have to assign it like so with this sign. All right, let's talk about sorting the table. Let's say that I want to sort the table by the petal length in decreasing order. This is what the order function does. So in this case, I am sorting the order of the rows by petal length. So let's see what that does. Now, here's your petal length, and it's in decreasing order. And now you note that the species is no longer sorted because you've sorted by a different variable. But let's say you wanted to sort by petal length within a species. So you would first sort by species and then by petal length within that. And R knows how to combine these two. So now you have, the petal length here by species, and this time it's increasing order. So it's going to go within Setosa, and then versicolor, and then virginica. If you wanted the petal length to be within decreasing order, then you would just add this negative sign before it. So this would do the same thing, except now the petal length would be decreasing. So for example, virginica, we see that it's decreasing order now. All right. Let's learn how to filter a table. Let's say I only want to get the rows that have a sepal length bigger than 7. So I would subset this table to only rows that have a sepal length larger than 7. See what that does. We now have a very small table, and all of these are bigger than 7. What if I only wanted to get the Setosa out of this table? So I filter by all the rows that have a species equal Setosa. All right, now it's only Setosa. Okay, now we saw in Excel that you had all these functions up here. You can sum, average, count, etc. So how do you do that in R? There's a function that was mentioned in the last lesson called colsums. That just gives you the sum of every column. Now, in this database, just to remind you, in this data set, if we look at the console, there are 4 numeric columns and 1 column that is a factor, or it really is a string. You cannot perform mathematical functions on a string, obviously. So you have to perform it only on numeric columns. So you actually have to exclude this last column from your analysis. Let's see actually what happens if you don't. So I'm going to be down here in the console, and I'm going to start printing the name of the function colsums, and then I can use tab to complete it. And I'm just going to write iris. And that throws in an error, which really makes sense, that says X must be numeric. X is the data frame. That means that you have a column in there that's not numeric, which is the name of the species. So in order to exclude that specific column, we take all of the columns from the iris database except for the last one. And the way to refer to the last column is by using the ncol function. So you could also just put a number in here if you know exactly how many columns you have in your data frame. But if you don't want to think about it, ncol iris will give you 5. So we're going to remove column number 5. And now you can actually perform this mathematical function. So this is the sum of all the columns, and let's do the same for the sum of all the rows. You can do the same thing to get the mean value for each column or row with the function colMeans. And again, you have to exclude the non-numeric columns. So here is your mean